Our next presenter is from the University of Alberta, Emanuele Goes, and she is going to present on the effects of chitosan oligosaccharides on broilers challenged with a natural subclinical necrotic enteritis model. Emanuele, please go ahead.
Thank you so much, Brenda, for the nice introduction, and good afternoon, everyone. So I will start with my introduction. So the ban of in-feed AGPs in animal diet has increased the susceptibilities to subclinical and clinical diseases. One of the most challenging diseases that we are facing in the field nowadays is the necrotic enteritis. The necrotic enteritis is an enteric disease caused by the pathogenic Clostridium perfringens, usually in the presence of predisposing factors. The necrotic enteritis can cause losses estimated in $6 billion per year in the mondial production. So some alternatives to reduce or minimize those losses would be to find a cost-effective replacement able to keep the animal's performance while minimize subclinical and clinical diseases, and also that can generate a safe protein source for human consumption. In the past decade, several products have been studied. However, there is still a lack of consistency and the results seem to vary from farm to farm. So in this context, we are studying the chitosan oligosaccharides, or COS, as a potential AGP replacement. So the COS can be originated from the degradation of chitin, and the chitin can be obtained from the shell of crustaceans, exoskeletal of insects, or from fungal cell walls. However, the chitin has a restricted application because of its complex molecular structure, mainly due to the high acetylation degree and molecular weight. However, when you transform the chitin into chitosan oligosaccharides, or COS, we can have a much simpler structure due to the removal of the acetyl groups, minerals, and proteins that could be attached to this molecule, and also because of the reduction of the molecular weight. So the COS is mainly formed by units of D-glucosamine, and the simple structure provides to the COS a huge pharmaceutical application, not only pharmaceutical, but also in the agriculture. The high molecular weight of COS is more related with antimicrobial properties. The medium molecular weight are more associated with the modulation of the immune system, while the low molecular weight is more associated with prebiotics properties. So in this context, the main objective of my project was to evaluate the dietary shellfish chitosan oligosaccharides of varying molecular weights as potential AGP replacements on broiler challenge with a natural subclinical necrotic enteritis challenge model. To achieve our objective, we fed our birds 8 different treatments. We had a positive control diet that was a commercial type diet with antibiotic and coccidiostat, a negative control diet that was a commercial type diet without any medication, and 3 other different COS products with different molecular weight and different levels of inclusion in the diet. From this experiment, we took these 2 specific treatments. The COS 95 KD included at 0.2 and 5 grams per kilo of the diet for further investigations. Because we selected these 2 in specific because they were able to partially restore the performance reduced after our challenge model. And I present these performance results in the meeting of the last year, so I won't touch much in those details, but this was the reason why we selected them for further evaluation and their effects on short-chain fatty acid production and the fecal microbial relative abundance. So to test the efficacy of our products in prevent necrotic enteritis, we applied a natural subclinical necrotic enteritis challenge model. And to induce the necrotic enteritis, we selected 2 predisposing factors. The first predisposing factor, we applied 15 times the recommended doses of coccidios vaccine by gavage at 12 days of age. And the main purpose of this first predisposing factor was to cause the first damage to the epithelial layer and then serve as entry point for the Clostridium perfringens proliferation. And 6 days later, we applied 24-hour feed removal at 18 days of age, and this served as a stress factor also to facilitate the proliferation of Clostridium perfringens. Since the Clostridium perfringens is ubiquitous, we expected that our birds had a natural uptake of the strains, the pathogenic strains and the other strains that is already present in our environment, in the litter and the feed and so on. So the main objective of this natural approach was to mimic the field conditions. In a recent publication, we characterized this challenge model and we validated this challenge model. And one of the most interesting things that we found was that this natural approach follows the natural progression of the disease induction. So this challenge model could be a good tool to cause subclinical necrotic enteritis in broilers, and also to test potential AGP replacements when the focus is to prevent necrotic enteritis. So at 22 days of age, we euthanized 2 birds per replicated, and we collected the total cecal content for the detection of Clostridium perfringens and its pathogenic strains by qPCR. We also analyzed the short-chain fatty acid concentration by gas chromatography and the bacterial relative abundance using 16S RNA sequencing. For the statistical analysis, the short-chain fatty acid concentration, we analyzed this data by one-way ANOVA, and the bacterial relative abundance, we analyzed this data using Kruskal-Wallis. For bacterial relative abundance in specific, we compare the cost treatments with negative control treatment and cost treatments versus positive control. For the confirmation of our natural infection approach, we detected the presence of Clostridium perfringens in all the analyzed samples, as we can observe here in this graphic, and the presence of net B positive strains in most of the analyzed samples as well, which confirms that we had this bacteria in our environment and that they could cause the necrotic enteritis. The reason why we decided to detect the net B strains was because these strains are able to produce net B toxin that is believed to be the toxin most related to the pathogenesis of avian necrotic enteritis. So here we have the results of the short-chain fatty acids concentration, and you can observe that the positive control treatment was responsible for the highest concentration of propionic acid in comparison with COS95 KDA included at 0.2 grams per kilo of the diet. When you see the results for isobutyric acid, the COS95 KDA, the one included at 5 grams per kilo of the diet, had the highest concentration of this acid compared to positive control and negative control treatment. The isovaleric acid tended to be significant, and again, the COS95 KDA included at 5 grams per kilo of the diet tended to have a highest concentration of this acid in comparison to other treatments. So here we have the results of the relative abundance of bacterial groups. In this case, we are comparing negative control with COS treatments. And at the genus level, we can observe that the negative control treatment was responsible for the highest abundance of Clostridium sensu stricto 1 and Erysipelatous Clostridium. Bacteria from those groups are more related to this balance disbalance. And at the species level, we can observe that the negative control treatment had the highest concentrations of Lactobacillus and Gluviae, while the COS treatment had the highest concentrations of Lactobacillus aureus. In a study with humans, these authors found a similar results. So the COS supplementation prevented the bacteria from Clostridium group to attach to the host cells in the gut, while benefited those from the Lactobacilli group. So the possible justification for that is that the COS could serve as a carbon source for the bacteria that can digest this polymer, producing short-chain fatty acids as metabolite, and then creating an appropriate environment for the growth of those beneficial bacteria. And then by competitive exclusion, those bacteria can eliminate some potential pathogens. And another explanation is that the COS could function as a soluble decoys. It means that they can attach to the bacteria that are free in the lumen that could not attach to the host cells, and then facilitate the elimination of those bacteria in the feces, or in the case of the birds, in the excreta. So here we have also the results for the relative abundance of bacterial groups, but now comparing positive control treatment with cost treatments. At species level, we can observe that the cost was responsible for the highest concentrations of Lactobacillus aureus and Lactobacillus engloviae, and also for the highest concentration of a lactated fermenter bacteria. Those results are in accordance with previous experiments that I found in the literature, where the authors supplementing costs in the diet, they saw an increase in lactobacillus counts, and it occurs in vitro experiments, broiler chickens experiments, pigs, and also human experiments. So it seems a very characteristic property of the cost. So as a final consideration, although we are still adapting our challenge model, we can say that this natural approach was able to cause a subclinical necrotic enteritis in broilers, and it might be a useful tool to simulate field cases of necrotic enteritis, and also to be used in experiments where you want to evaluate AGP replacements. The COS95 KD may act as a prebiotic, modulating the growth of lactobacilli species in broilers. And as we saw like in previous meetings that I presented us, the COS95 KD could also improve the performance. So this modulation in the growth of beneficial bacteria might be related to enhancements in performance as well. And in addition to a prebiotic function, the COS may stimulate the biosynthesis of branched chain amino acids that are involved in the enhancement of intestinal integrity and also in the production of endogenous defensins. And the COS can also be recognized by immune cells. So the COS can modulate immune responses, mainly at the mucosal level. And all this together can promote for the host a better intestinal health. So it might be also an explanation why you found better performance results applying this specific COS. So as a final conclusion, we can say that COS95 KDA may be a part of a strategy to replace AGPs. However, complementary analyses are still needed to be performed, and further mechanistic studies are required before you make recommendations for the use of this product in the industry. So I'd like to thank our partners and sponsors of this project, and thank you so much for attention. I'll be happy to answer any questions you guys may have.
Thank you very much, Emanuele. That was very interesting. I have a couple questions for you, and just a reminder to everyone that if you have questions for Emanuele, to pop them into the Q&A box. The first question I have, it sounds like the challenge model that you've worked on could have some potential application beyond testing the chitosan oligosaccharides, the COS as you call them. Can you talk a bit about how it was developed or anything? I know you didn't have a lot of time to really talk about it, but can you just talk about how you think it could be applied and more about the model?
Sure, thank you for your question. So this challenge model is new. We are still adapting this challenge model, but you saw very promising results from the use and application of this challenge model. And we also did a recently publication characterizing this challenge model. So if someone is more interested in that, I encourage you also to read this paper. But when you thought about this challenge model, we were thinking about to simulate the field conditions as closer as possible. So in the case of our study, we want to see, to evaluate the potential of AGP replacements. So we want to test this AGP in a commercial or more closer to the commercial condition, right? So usually the conventional challenge models, the necrotic enteritis conventional challenge models, they apply to the birds a specific Clostridium perfringens bacteria for 3 or 4 days in a row, for example. But in the case of our experiment, or when you are trying to test the AGP replacement, you want to be closer to the field conditions. So in the field, the birds are in contact, or they uptake the Clostridium perfringens that are in the environment, right? So we are not providing those bacteria. I'm not saying that the conventional models are not useful, but it depends of the purpose of our study, the type of, the severity of the disease that we want to cause, and what you want to evaluate in those experiments. So in our case, you want to evaluate AGP replacements. We want to cause subclinical necrotic enteritis that is more mild. So we don't want to provide loads of bacteria and then cause clinical necrotic enteritis, because in the field, what really happens, like what the most common situation is to have subclinical necrotic enteritis that can reduce the performance of the birds without showing clinical signs. So usually the producers can only see that in the end of the cycle, right? When those birds are in the slaughterhouse, for example. So because of that, we are trying to apply and adjust this challenge model instead of other conventional ones, because for the purpose of our study, this challenge model seems a good and effective tool. Awesome. That answer your question?
Yes, it did very well. Thank you very much, Emanuele. We did have a question come in, and I think I know the answer to this, but I will ask it. The question is, the challenge model that you used caused clinical or subclinical forms of necrotic enteritis?
Okay, that's a good question. So we want to cause a subclinical necrotic enteritis. That's why you are very careful about the application of this challenge model, because we want to avoid clinical signs. And from what you have seen, we were able to cause this subclinical necrotic enteritis. And the way that you can make sure that we cause subclinical necrotic enteritis, since it's just reduced performance, is checking the lesions in the intestine in different periods. So usually what happens, because the subclinical decreased performance, is because it can impair the digestion and absorption of nutrients, because we have these lesions in the intestine. So if you are checking these lesions frequently during the experiment, we can make sure that we are causing a subclinical necrotic enteritis instead of clinical. Clinical, you see high rates of mortality. So it can reach 1% a day. So we don't want to cause it, because it's not the most common severity of the lesion that occurs in the field.
Okay, great. Thank you so much for your great answers to the questions and for your very interesting presentation.